Rhodopsin is a light-sensitive receptor protein involved in visual phototransduction. It is named after ancient Greek omicron delta omicron nu 4 rose, due to its pinkish color, and psi iota sigma 4 site. Rhodopsin is a biological pigment found in the rods of the retina and is a G protein-coupled receptor. Rhodopsin is extremely sensitive to light, and thus enables vision in low light conditions. When rhodopsin is exposed to light, it immediately photobleaches. In humans, it is regenerated fully in about 45 minutes. Rhodopsin was discovered by Franz Christian Boll in 1876. Structure: rhodopsin consists of a protein moiety also called scotopsin, which binds covalently a cofactor called retinol. Scotopsin is an opsin. Opsins are G protein-coupled receptors and have 7 transmembrane domains. The 7 transmembrane domains form a pocket, where the retinol binds to a lysine residue in the 7th transmembrane domain. The retinol lies horizontally to the cell membrane, and the cell membrane lipid bilayer embeds half of the rhodopsin. Thousands of rhodopsin molecules are found in each outer segment disc of the host rod cell. Retinol is produced in the retina from vitamin E, from dietary beta-carotene. Isomerization of 11-cis retinol into all-trans retinol by light induces a conformational change in opsin, continuing with meta-rhodopsin 2, which activates the associated G protein transducin and triggers a cyclic guanosine monophosphate second messenger cascade. Rhodopsin of the rods most strongly absorbs green-blue light and therefore appears reddish-purple, which is why it is also called visual purple. It is responsible for monochromatic vision in the dark. Several closely related opsins exist that differ only in a few amino acids and in the wavelengths of light that they absorb most strongly. Humans have 8 different other opsins besides rhodopsin, as well as cryptochrome. The photopsins are found in the different types of the cone cells of the retina and the basis of color vision. They have absorption maxima for yellowish-green, green, and bluish-violet light. The remaining opsin is found in photosensitive ganglion cells and absorbs blue light most strongly. In rhodopsin, the aldehyde group of retinol is covalently linked to the amino group of a lysine residue on the protein in a protonated Schiff base. When rhodopsin absorbs light, its retinol cofactor isomerizes from the 11-cis to the all-trans configuration, and the protein subsequently undergoes a series of relaxations to accommodate the altered shape of the isomerized cofactor. The intermediates formed during this process were first investigated in the laboratory of George Wall, who received the Nobel Prize for this research in 1967. The photoisomerization dynamics has been subsequently investigated with time-resolved IR spectroscopy and UV-vis spectroscopy. A first photoproduct called photorhodopsin forms within 200 femtoseconds after irradiation, followed within picoseconds by a second one called bathrodopsin with distorted all-trans bonds. This intermediate can be trapped and studied at cryogenic temperatures, and was initially referred to as prelumirodopsin. In subsequent intermediates lumirodopsin and meta-rhodopsin I, the Schiff's base linkage to all-trans retinol remains protonated, and the protein retains its reddish color. A critical change that initiates the neuronal excitation involves the conversion of meta-rhodopsin I to meta-rhodopsin II, which is associated with deprotonation mutation of the Schiff's base and change in color from red to yellow. The structure of rhodopsin has been studied in detail via X-ray crystallography on rhodopsin crystals. Several models attempt to explain how the retinal group can change its conformation without clashing with the enveloping rhodopsin protein pocket. Recent data support that it is a functional monomer instead of a dimer, which was the paradigm of G-protein-coupled receptors for many years. Phototransduction. Rhodopsin is an essential G-protein receptor in phototransduction function. Rhodopsin 2 activates the G-protein transducin to activate the visual phototransduction pathway. When transducin's alpha subunit is bound to GTP, it activates cGMP phosphodiesterase. cGMP phosphodiesterase hydrolyzes cGMP. cGMP can no longer activate cation channels. This leads to the hyperpolarization of photoreceptor cells and a change in the rate of transmitter released by these photoreceptor cells. Deactivation method 2 is deactivated rapidly after activating transducin by rhodopsin kinase and arrestin. The rhodopsin pigment must be regenerated for further phototransduction to occur. This means replacing all transretinal with 11-cis retinal, and the decay of matter 2 is crucial in this process. During the decay of matter 2, the Schiff base link that normally holds all transretinal and the apoprotein opsin is hydrolyzed and becomes meta 3. In the rod outer segment, meta 3 decays into a separate all transretinal and opsin. A second product of meta 2 decay is an all transretinal opsin complex in which the all transretinal has been translocated to second binding sites. Whether the meta 2 decay runs into meta 3 of the all transretinal opsin complex seems to depend on the pH of the reaction. Higher pH tends to drive the decay reaction towards meta 3. Rhodopsin and retinal disease. Mutation of the rhodopsin gene is a major contributor to various retinopathies such as retinitis pigmentosa. In general, the disease-causing protein aggregates with ubiquitin in inclusion bodies disrupts the intermediate filament network and impairs the ability of the cell to degrade non-functioning proteins, which leads to photoreceptor apoptosis. Other mutations on rhodopsin lead to X-linked congenital stationary night blindness, mainly due to constitutive activation. When the mutations occur around the chroma-4 binding pocket of rhodopsin, several other pathological states relating to rhodopsin have been discovered, including poor post-Golgi trafficking, dysregulative activation, rod outer segment instability and arrestin binding. Microbial rhodopsins. Some prokaryotes express proton pumps called bacteriorhodopsins, proteorhodopsins, and xanthorhodopsins to carry out phototrophy. Like animal visual pigments, these contain a retinal chroma-4 and have 7 transmembrane alpha helices. However, they are not coupled to a G protein. Prokaryotic halohodopsins are light-activated chloride pumps. Unicellular flagellate algae contain channel rhodopsins that act as light-gated cation channels when expressed in heterologous systems. Many other pro- and eukaryotic organisms express rhodopsin ion pumps, as sensory rhodopsins of yet unknown function. Very recently, a microbial rhodopsin with guanylyl cyclase activity has been discovered. While all microbial rhodopsins have significant sequence homology to one another, they have no detectable sequence homology to the G-protein-coupled receptor family to which animal visual rhodopsins belong. Nevertheless, microbial rhodopsins and GPCRs are possibly evolutionarily related, based on the similarity of the three-dimensional structures. Therefore, they have been assigned to the same superfamily in structural classification of proteins.